My name is Belle Shenoy. Let me give some information about myself. Um, I'm a co-founder and the managing editor of MycoAsia journal. Presently working as principal scientist at CSIR National Institute of Oceanography in Visakhapatnam, Andhra Pradesh, which is on the east coast of India. I did my PhD from the University of Hong Kong in 2007 on the topic of fungal taxonomy. After that, I did my postdoc in Agriculture Canada, Ottawa. Since 2008, I have been working for CSI labs in India. I do research on fungi and bacteria. I teach, especially PhD students. I edit for journals and I promote mycology using social media tools. This is my email address and this is my WhatsApp number, and this is the website of MycoAsia, www.mycoasia.org. Today I will be talking about Mega. So what is Mega? Mega is a computer software, as you may know. It's a computer software. The full form of Mega is Molecular Evolutionary Genetics Analysis. Okay. What does Mega do actually? Mega conducts statistical analysis of a molecular evolution. If you have a data set of DNA or protein, you can conduct statistical analysis of a molecular evolution. Mega constructs phylogenetic trees. What is a phylogenetic tree? I will explain in coming slides. This is the website of Mega Software, www.megasoftware.net. So these are the original authors of the Mega Software. This software is being developed by Pennsylvania State University in the USA. It was initially released in the year 1993, almost 29 years ago, and presently the 11.0.11 version is available on the website. It is a freeware, proprietary freeware. You can use it for free. Therefore, I am teaching you Mega today. Let me go to the website of Mega and show you some things. This is the website of Mega. Okay, from here you can download the software. Here are certain things you can consider. Here is a tutorial button for videos, walkthroughs, books and articles, and next button will give features of Mega. And if you are interested to read documentation related to Mega software, you can go to this button, tab actually. If you want to give any feedback to the authors, you can use this feedback button. Okay. If you see here, Mega has already been downloaded around 2.9 million times. Okay. So it is already being, used by researchers from all over the world, as you see here actually. And you see Indian and some Chinese, Thai people and some Indians are active right now. Why I am teaching Mega? I mean, there are many other programs, but why I am so focused on Mega? I teach Mega because it is easy to use. Well, it's just about click, 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 and it is best suited for beginners, and it is free to use, as I told you earlier. And since 1993, it has transformed with new features actually. Therefore, it's quite effective with all the tools now. Now let me give some understanding about phylogeny actually. Like an organic tree which has a root, okay, and the stem and leaves. Phylogenetic tree has a root and it has branches and finally whole leaves here actually. So if you see a tree of life here, you have animals here, you have fungi here, you have plants here, and prokaryotes somewhere here. Okay. The science of classifying, naming, and describing organisms is known as taxonomy. It's a science. Taxonomy is a science actually. When we describe a new species or a new genus or a new taxon, it is a hypothesis. So a hypothesis is supported with set of data set or data. So tomorrow someone else can prove or disprove it actually with the additional set of data. So taxonomy is a hypothesis-centric, hypothesis-based science, and taxonomy has three components. It has identification, classification, and nomenclature. So in identification, Identification is a binary process. In identification, if it is determined whether a particular individual belongs to the known groups of organisms or not. Okay, if it doesn't belong to any known groups of organisms, then you have to create a new taxon, new category for it actually. So now you can classify organisms based on similarity. Similarity at the level of morphology or at the level of gene sequence data. It can be based on polyphasic data. Okay, so classification is hierarchical, as you know, starts from species, genus, then order, family, order, etc., up to the level of domain actually. So the nomenclature governs the naming of organisms. So you cannot, you can call an organism by a common name, but the scientific names follow nomenclatural rules actually. So as you may know, modern taxonomy includes phylogeny as an integral part of the classification process. So phylogeny is the part of classification process. Okay, what is phylogeny? Phylogeny is nothing but evolutionary relationships of organisms and genes. So the basic hypothesis or basic funda behind this is that genes and proteins acts as evolutionary chronometers or evolutionary clock. So a clock measures the evolutionary change here. Therefore, genes and proteins have been used as markers actually. So there are certain requirements to be a good marker. First of all, that marker should be present in all members of the target group. It should be functionally homologous in the organisms. And that marker should contain both conserved region, because conserved region helps in the alignment of the data set, and just conserved regions will not be enough, because you need phylogenetic signals, and the phylogenetic signals are there in variable regions. So these variable regions should be slow enough to permit the measurement of evolutionary change. These are the markers used for molecular identification. For animals, cytochrome oxidase. For plants, maybe RDCL and ITS. For bacteria, 16S or RNA. For fungi, ITS. For protists, ITS. So this is from Wikipedia page. Okay. A standard molecular phylogenetic analysis flow starts with an organism. Okay. In this case, a fungal culture. You would like to keep a copy of the fungal culture for long-term preservation, and another copy for the genomic DNA isolation, then the PCR and sequencing of desired genes. In case of fungi, maybe ITS to start with. Then you do once you get the sequences, you do the phylogenetic analysis. Then the phylogenetic analysis results in phylogenetic trees. So what is a phylogenetic tree? Phylogenetic tree is nothing but the graphical representation of evolutionary relationships. It is nothing novel in terms of concept. Even Charles Darwin used the concept of phylogenetic tree in his notebook. So this is from Darwin's notebook. In the modern era, you have a phylogenetic tree of life. Phylogenetic tree of life. It has got 3 domains: bacteria, archaea, and eukaryotes, and here you have a hypothetical ancestor here. So what are the parts of a phylogenetic tree? So phylogenetic tree has a root. Okay, that is the oldest ancestor of all the taxa. Then you have internal nodes. Then you have terminal nodes. So internal nodes represent the hypothetical ancestors of these taxa actually. In this case, this node represents the hypothetical ancestor of these 2 taxa. These are the present day organisms actually, or genes or populations of species. Okay, then you have branches. Branches gives an idea about the amount of evolutionary change. Okay, so I'm repeating, a phylogenetic tree has a root. The phylogenetic tree has internal nodes. The phylogenetic tree has terminal nodes. Okay, then the branches are connecting these nodes, and the branches gives an idea about the evolutionary change that might have occurred. How to construct a phylogenetic tree? For constructing a phylogenetic tree, you need to have a data set. In case of fungi, you can start with ITS data set. You can have your own sequence, okay, or you can borrow from public databases. Once you have a sequence of desired number, once you have a desired number of sequences, say 50 sequences, 100 sequences, then you have to create a multiple sequence alignment. That you can do it in Mega, once you create a multiple sequence alignment, you have to find out the best tree or best trees using tree building methods. So once you get the best tree or best trees, you have to evaluate that branching pattern for statistical support. That is done by bootstrapping. Finally, once your tree is there with bootstrap values or statistical support values, you have to present in a very professional manner, in a scientific manner. Now coming to the assembling a data set, okay, you can have your own sequences. When you have your own sequence, you need to get homologous sequences for public databases. The best example is NCBI, okay, and Treebase and Easy Taxon. When you go to NCBI, there are two basic kinds of search strategy. First one is you type the name of the organism, then you will get the results. Or you go to NCBI BLAST option, you paste your sequence, you get homologous sequences. So this is a multiple sequence alignment. I mean, basically a picture of it. Here each column is a character. Okay, each column is a character, and these different nucleotides are character states. Here C and A are character states. So these differences are indication of phylogenetic signal. These are the phylogenetic signals. So this is a column with only T. This is a character with only one character. There is no character state, only one character there. So this may not have very rich phylogenetic signal. Okay. Here you have a column with A and dash. Dash can be missing data or an insertion. Okay. So this is a column, a character with two character states, A and dash, missing data. But these columns may be misleading sometimes. Okay, so you sometimes would like to delete these kind of things. You can use Mega software to create a multiple sequence alignment. Okay, or I used to use CView also. If you have more than 500 sequences, maybe 300, if you have a large data set, probably then you can use MAFT server. You just upload the MAFT FASTA file onto the server, and the server will give an alignment file, multiple sequence alignment after some time, depending upon number of sequences you have added in that file actually. So in a multiple sequence alignment, every residue from every sequence is homologous. Homologous is a hypothesis that you can adjust by eye to minimize insertion deletion events. Normally DNA sequences are used for closely related sequences, and amino acid sequences are used for distant relationships. This is creating a multiple sequence alignment is is an important step. Okay, if you are not careful, if you are immature when you create a multiple sequence alignment, immature in the sense taxonomically immature, you might get a multiple sequence alignment, but the tree tree that would be created based on that would be a garbage. So garbage in is garbage out when it comes to multiple sequence alignment. Alright, there are two categories of tree building methods. So tree building methods, what do they do? These tree building methods find out the best trees from the multiple sequence alignment. Actually, there are two tree building methods: distance matrix methods and discrete data methods. Okay. In the distance matrix method, there are two examples: neighbor joining. This is quite popular. UPMG, not that popular nowadays. Among discrete data methods, maximum parsimony, maximum likelihood Bayesian. These are the best examples. Let me give more information about tree building methods. In case of distance matrix method, okay, let me explain again. There are two categories of tree building methods: distance matrix methods and discrete data methods. Okay, and in a distance matrix method, which a multiple sequence alignment is first converted into a data matrix. That data matrix is used for the construction of the phylogenetic tree. Okay. In case of distance matrix method, for example, neighbor joining, a multiple sequence alignment is first converted into a data matrix. That data matrix is used for the tree construction. But in case of discrete data, which includes parsimony, maximum likelihood, Bayesian methods,
Multiple sequence alignment is not converted into a data matrix. Instead, it is directly used. This multiple sequence alignment file is directly used to construct the tree. So this data matrix step is not there in discrete data. Once you get the phylogenetic tree, so you need to find out the branch, statistical support for the branching patterns. Okay, that is done using bootstrapping. In bootstrapping, there's the mega software. For example, take random subsamples from multiple sequence alignment, build trees from these subsamples, and calculate the frequency of occurrence of different parts of the tree. For example, 89.7% means out of 100 times, 89.7 times this branching pattern is supported. Okay, so there is a chance that 11 times it may be wrong. Okay, it may not be supported. So basically, bootstrapping tests the phylogenetic accuracy of the tree. Bootstrap values 70% or higher are likely to indicate reliable groupings. Okay. If there is no support or less support, less than say 70% support, the branch will collapse, not literally but figuratively. Okay. If you are interested, you can go to YouTube and watch this video on bootstrapping. It's 11 minute long, so just watch it leisurely, and you'll get an idea about bootstrapping. Finally, the data presentation. In data presentation, branch lengths are drawn to scale. Okay, so longer the branch length, the higher the amount of evolutionary changes that might have occurred. Actually, branch length, as I told you, it gives an idea about the amount of evolution estimated to have occurred. Bootstrap value should be displayed in percentages here. Okay, bootstrap values 50% or higher are reported. Bootstrap values less than 50% are not reported. You can take one of the divergent sequences, divergent to this your target group. This sequence here, for example, is used in an out group, and this sequence is used for rooting of the tree. Okay. So what happens when you root a tree? When you root a tree, it gives an idea about the direction of the evolution. What is meant by direction of evolution? It gives an idea about which is the recent branch, which is the not so recent branch among these lineages. Okay. These are the three important papers. You might have seen this paper, Phylogeny for the Faint of Heart tutorial. This is for the beginners. If you like complicated stuff, you can read this Nature Genetics Reviews paper. This is for the step-by-step methods for tree building. Okay, maybe this is a little bit outdated. Okay, let's start downloading the software. Okay, for downloading the software, we'll go to MycoAsia. Accept the terms and conditions. I'm from India. I'm a researcher from CSIR, India. I'm from a research institute. Then say download. I will execute the file. I accept the agreement and I say next. Okay. Finish. So here is your mega software.
Okay, now now I take an example file. Just watch me construct a tree from an example file. Okay, here is the folder. Okay, I click on that. Then I select the fourth file, command underscore statements.meg. Click on that. I would like to have a look at the data set. Okay, I'll just have a look at it. Okay, you can have a look at this. See, these columns just dot dot dot. These are constant character actually. Okay, this column, this character is two character states. Here A, he has got A and dot. So this character has two character states. Okay, as I explained before. Just have a look at it and then minimize it. Okay, this will be here. This is an active data set. After that, go to the sixth button, sixth option. 1, 2, 3, 4, 5, 6. Phylogenetic. Okay. Click on it. Then select the second option. Construct or test neighbor joining. Click on that. Would you like to use the currently active data? Yes, this is the currently active data. Yes. Here, test of phylogeny should be bootstrap method, which I explained during my presentation. You select bootstrap method, number of bootstrap replication, I would prefer 100. You can go up to 1000, I guess. Then you say OK. Now you see the tree. I take the last sequence as an outgroup. To do that, I click on the branch. You can see it is highlighted now, the green rectangle. Then I right click on it, then root the tree. So this is the rooted tree. Now you go to image, copy to clipboard, then open your paint. Then paste it, Control V, or you can go to somewhere here, view, I think not sure where, Control V. Then save the file as JPEG file, my tree 2. Save the file. In the next activity, I am going to show you how to go to NCBI website, select some sequences and trim some information related to the sequences. Then create a multiple sequence alignment. Then create a neighbor joining, construct a neighbor joining tree, construct a phylogenetic tree, then do the bootstrapping, then root the tree, take the screenshot. First I go to Google. I type NCBI. Now you click on nucleotide. Then I'm typing the name Aspergillus. This Aspergillus is a fungus, Aspergillus niger. ITS is the gene region, and I want sequences only from the type material. If you don't know what is meant by type material, Google it. Now I say search. And NCBI is suggesting something. I click on that, Aspergillus niger ATCC 16888 ITS region from type material. I click on that. I can see all the metadata here. That's okay. Then I click on run blast. Okay. Click on that. The sequence, the accession number is selected. Okay. I click select sequences from type material. Then here somewhat similar sequences. And I click on blast. Okay, this is the result from the blast. First I uncheck. Okay, then I check, select this. This is our sequence. Then I select the first 5 including mine, our query. Then I select one divergent sequence, maybe this one, with the 95% around 96% sequence identity. So totally 6 sequences will be downloaded from this download section, and I'll go for aligned sequences. So a file will be is being downloaded here. So these are the sequences. I select all the sequences. And I open a Word file. Paste my sequences. I will use the option Control V. Okay, the sequences have been pasted here. Decrease the font size for visibility, better visibility. Then I remove some of the things here. These are redundant information. ITS from type. I delete that. This is strain number. I will keep it. This is species name. This is genus name, and this is gen bank accession number in the region which is retained. Okay, I will delete here these things because it conveys the same thing, that is an ITS sequence. We know all our ITS sequences. Just a tip, okay, just click on this. Make sure there is a space between this sentence and this line and the second line. So this space should be there. Otherwise you may face some problem. Okay. Now select these 6 sequences. Open Mega. Go to alignment, align, select edit or build alignment. Here select create a new alignment. Okay. Are we building a DNA or protein sequence? DNA sequence data. And right click on this and say paste. Remove the empty sequence, empty entry here. And edit, select all sequences. All sequences have been selected. Then you go here, alignment, below alignment, align using Muscles algorithm. Click on that, align DNA, and so you say OK. Those sequences are aligned now. Okay. Now you go to data, phylogenetic analysis, protein coding nucleoside and nucleotide sequence data, no. You go to phylogeny, select the second one, construct and test Phylogenetic tree, yes. Test of phylogeny bootstrap method, number of replicates 100, all the remaining things are default settings. So my tree is here. This is my outgroup. Just click on it, double click, right click and go tree. Then I go to image, copy, or I go to image, copy, go to paint, and paste, Ctrl V, file, save as, JPEG tree, my as per the tree.